a novel method for learning sample-level representations with clinical applicability across diverse patient populations. Welcome.
Hello. Welcome to my presentation. Imagine you have a diagnostics tool that is approved for a certain group of patients, but limited to a certain age group. Now you have a new patient coming in that demands this diagnostics tool for a disease, but he exceeds this age limitation. As a practitioner, you would have to turn this person away, but I think we can all agree that we would like to know if this person might be eligible for this diagnostics tool. Now, in recent years, we've seen sort of an explosion in technologies and datasets in the single-cell field, where we now have datasets reaching hundreds of millions of cells, where we capture thousands or tens of thousands of genes for each of these cells. So we have huge and largely dimensional data. However, in these datasets, we mostly capture differences in cellular states. When we're thinking about clinical applicability, usually clinical conditions manifest at sort of an organ or system level, and this is also where we typically make clinical decisions. So we need to find ways to bridge the scaling gap from single cell to the patient level, and this is exactly where sample representation methods as a sort of new subfield emerges. Applications for these sample representations range from diagnostics, prognostics, over patient stratification to biomarker discovery, or disease trajectories. We have a toolbox called PadPy for this if you want to learn more about this or apply this to your data. Now, taking a step back and formulating this problem in a bit more machine learning way, we can understand a sample as a bag of cells of varying cell types or states and varying number. A sample representation can then be understood as a mapping of this input set to a singular vector zi, here denoted, which we would understand as a sample representation. An important property of these functions is that these functions need to be permutation invariant, meaning that this learned mapping should not depend on the ordering of cells, neither on the number of cells. Now, in this emerging field, there have been first approaches been published recently. I mean, the simplest one doesn't have to be published, is simple pseudo-bulking, but there's also work even towards foundation models in this field. However, most of these approaches have sort of limitations like label dependencies or batch effects. So we thought, how can we tackle this problem and going away from a certain label dependency? And we ended up with thinking of this from a contrastive learning self-supervised point of view. Now, we took inspiration from the computer vision world, in particular from the SampleCLR algorithm, which is a fairly simple, I think, quite elegant approach where we learn embeddings or representations by creating different augmentations of images, and then via specific loss formulation, trying to pull together similar augmentations from the same image while pushing away dissimilar ones. Took a similar idea for single cell. We take a patient, we create non-overlapping subsets of cells of these before mentioned bags, and then we learn this contrastive way to pull together the same patient while pushing away dissimilar patients. For our method in particular, how we approach this is that we define a set of heads, so-called aggregation heads, where we feed in these subsets of cells and sort of learn different weights for the different cells. In the next steps, we combine the heads, project them down into a latent space, and apply our contrastive objective. So really learning without the need for label guidance. For the loss formulation, we've selected the XCLR loss, which is a variation of a commonly used info-NCE loss, which further allows us to input prior information on sample similarity. In addition to this, our method is quite modular, so for anyone using it, you can add sort of supervised heads that allow you to further guide the model with clinical covariates that you might be interested in. In order to assess this, and in our lab we like benchmarking a lot, we performed a quite large-scale benchmarking where we compared essentially against all existing sample representation methods. It's a quite new field, so there's not so many methods out there, but nevertheless, we tried them all, and we calculated these metrics across a wide range of different data sets, across tissues, conditions, and a lot of donors. So each dot in this scatter plot you can see here corresponds to the results of one method across all of these data sets, and we measure both the information retention and trajectory preservation. These two scores assess how well we can recapitulate certain clinical labels and essentially measure how well we can predict clinical. You can see that we have sort of a clustering in this scatter plot, where on the top right, the best performing models are supervised models, and here our methods sort of beat the current state of the art. In the other clustering, we have mostly unsupervised and first foundation model methods, and also here we reach state of the art in the information retention and are as good as the best in the trajectory preservation. Now, anyone that's been working or has worked on biomedical, or I guess also other data domains, will know about the issue of batch effects. Batch effects sort of arise when you have technical signal from, for example, different experimental protocols, sort of overshadowing the biological signal you want to capture. Especially in clinical applications, this is problematic because we want to capture disease signal and identify treatment opportunities. So we need to find a way to sort of account for these. Previously, there has not been a lot of, or basically no method that explicitly tackled this issue in the sample representation field. There's a lot of work in the single cell field and really well-working methods, but we thought, how can we approach this? So what we do is we don't go via like an explicit loss formulation or forcing the model to ignore batch effects, but we do this sort of an implicit way, where we create training mini batches for the model. So if you think about how we train these deep learning models, usually we train per mini batch and we gradually update our weights. So what we do is we keep only one experimental data set at a time, and then we gradually fill up these mini batches with similar data points, really enforcing the model to only focus on one batch at a time, while also making sure that we learn meaningful representations. Now, we again compared this to all the other methods out there on a batch correction score, and we showed that not only did we improve the batch the batch effects, but we did so very well. And what I want to show you here is that when we apply this batch correction, we really see that we drastically improve the score, leading to meaningful representations. Now next, we were interested in how scalable our model was and looked into a range of data sets. So from left to right, we have increasingly large data sets with increasingly many patients. And with the stars here, you can see it's denoted here data sets that are GPU, or methods that are GPU accelerated. And we can see that our method here, highlighted in the color, scales quite nicely. Actually, it scales quasi-linear with respect to the number of samples. And especially in the larger data set here on the right, we saw that compared to the current best existing methods, our model is much more efficient. Now, coming back to the initial problem I mentioned about, which is clinical applicability and what we are fundamentally interested in, I'll present you a use case here. So we looked into a combination of two large existing COVID data sets, which are known to have quite technical differences. We then trained our self-supervised batch-aware sample representation method and further guided the method on sort of classifying disease. For downstream, we then looked into certain disease trajectories, as well as if we could identify biomarkers. So first, I want to show you the latent representation of our method, where we can see somewhat nice separation of the diseases, indicating sort of as proof of concept that our model meaningfully captures disease signal. We then looked into the pseudotime, sort of an axis along the severity of the diseases, and we found a gradient indicating meaningfulness in what our model actually learns. Looking at this from a slightly different perspective, we can see here the severity of the diseases, so different labels of disease severity, on the y-axis, and then the pseudo-time along the severity along the x-axis. And what you can see is that our model is able to sort of capture a pseudo-time, like a gradient in the embedding along this disease trajectory, which is a really nice finding because this essentially allows us to, in a future case, map in new patients and sort of map them onto this trajectory and assess their severity status. We went one step further and looked into underlying cell types and programs, and whether we could recapitulate any disease-associated cell programs. And we found that certain cell types that are known to be correlated or important in COVID pathways were highly correlated to our pseudotime, again indicating that our model is able to meaningfully capture this disease signal and severity trajectories. So to summarize, I showed you SampleCLR, our new patient representation method, and showed you how we outperform most existing methods across different conditions and tissues at orders of magnitude fewer parameters. To our knowledge, we're the first sample representation method to do so in a batch-aware manner, so keeping biological signal while ignoring technical artifacts. We do all of this while maintaining an interpretable, lightweight model with a scalability, modularity, and we envision this to be a step towards true foundation model scale sample representations. Finally, we show clinical applicability across disease prediction, biomarker discovery. And with this, I want to end and especially thank everyone involved in the project and the lab.
Thanks a lot for the presentation. Are there questions? Yes, in the front here. Can you maybe raise your hand again so the people with the mic?
Thank you for your talk. Back to the clinical applicability, do you think or do you know if the quality of the diagnostic test, so the sensitivity and the specificity, impacts the performance of the model downstream? So, or how the data is generated upstream? Yeah.
Yeah, I think it's a super good question. So we mostly work with like large-scale single-cell data sets, and for the clinical sort of impact, or like the clinical things that we look into, we mostly rely on like existing metadata. So it really depends very much on how well the data, or people in the lab that have generated the data, how confident they are and how well this works. But if, let's say, the experiment is conducted nicely, then like our model doesn't add like a layer of sort of uncertainty to this. So it's basically just learning a representation of this data to simplify these predictions. Yeah.
Okay. I think Fabian has a quick question.
Hi, Benjamin. Interesting. So what's missing towards sort of a foundational type of work there? There have been first ones with Pulsar or whatever, right? I mean, they sort of threw a bunch of data sets together. Realistic to do the same with your approach?
I think it's very realistic, and to me it seems, Pulsar's, I think, a really cool paper. Our tests perform so-so. I think like the data, this like quality and size of these data sets, like really having like a lot of patients with like good clinical annotations is like the step that is maybe missing towards like this notch of.
I don't think you need clinical annotation for just the representation, or you just need the badge.
Yeah, for the representations you can learn them. I mean, yeah, but I think
again, that's I think.
Yeah.
Then you know the use cases can be smaller, but for the annotation you just need batch information, and that's what you have, right?
Yeah, yeah.
And thank you again for the presentation. And